welcome to today's session on Prostaglandins. Myself, Dr. Mausmi Shankar Adala, working as an assistant professor in the Department of Biochemistry, St. Joseph's College for Women, Autonomous. The contents included in under this topic are eicosanoids introduction, prostaglandins, nomenclature, pathway, and structure.
Learning outcomes will be all prostaglandins, pathway, and its role.
Eicosanoids are lipid fatty acids with 20 carbons that have biological activity. Eicosanoids belong to the class of paracrine hormones and are bioactive hormones with local action that act close to the site of hormone synthesis illness. Arachnoic acid and related polyunsaturated acids, abbreviated as PUFAs, including eicosanopentanoic acid EPA, are the sources of eicosanoids. In the diagram, we can very clearly see what are prostaglandins E1, thromboxane A2, leukotriene B4, prostacyclin I2, and leukotriene. All these are derived from your parental compound, that is nothing but eicosanoids. The four major classes of eicosanoids are as follows: prostaglandins, PGs, LTs, lipoxins, LX. The prostaglandin the prostate gland produces prostaglandins, which typically have a localized effect on surrounding tissues. They quickly break down into inactive compounds at the site of action, making them unstable. As a result, prostaglandins are not significantly concentrated in blood. Prostaglandins have a variety of biological effects, such as promoting the uterine smooth muscle contraction, altering the blood supply to particular chain are represented by nimbus. Prostaglandins and thromboxanes are referred to as prostanoids together. The number of carbon-carbon double bonds is indicated by a subscript number in the nomenclature of prostanoids. Because of majority of prostanoids containing two carbon-carbon double bonds, they are referred to as series of two molecules. Arachnoic acid is the source of series 2 prostaglandins, while EPA is the source of series 3 prostaglandins. With 4 carbon-carbon double bond, series 4 molecules are the most prevalent type of leukotrienes because they are 4 double bonds between carbon atoms. Biological functions serve a variety of biological purposes. That is a primary role, which are in the regulation of blood pressure, stomach secretion, reproduction, and inflammation. Additional homeostatic roles played by eicosanoids include controlling platelet aggregation, mucosal integrity, and regulating vascular leakage. For example, the metabolite TXA2, which is produced from COX-1, plays a significant part in the platelet aggregation. Conversely, inhibits platelet aggregation and encourage vasodilation. Synthesis of eicosanoids. Arachnoic acid 20 carbons is the main precursor of prostaglandins and the other related eicosanoids. The major component of phosphodienol inositol and the other complex lipids forms is arachnoic acid. Phospholipase A2 is a primary enzyme responsible for the release of free arachnoic acid, which is linked to phospholipid. Other acyl hydrolases may be involved. Hormones and other stimuli regulate the help of the enzyme. Phospholipase A2 produces arachnoic acid. Whereas the same diacylglycerol in the presence of phospholipase C also produces arachnoic acid. This arachnoic acid, under the influence of lipooxygenase FLAP or ALOX5 enzyme, it is converted into HPETE, which is nothing but hydroperoxy eicosanoic acid. Also, the same arachnoic acid in the presence of PGH2 synthase, that is COX-1 or COX-2 and peroxidase, is converted into prostaglandin H2, PGH2. Now, this prostaglandin with the help of PGA synthase can be completed into PGE, which is further interconverted into PGF2, or else it can directly be converted into PGD2 with the help of PGD synthase. Now, prostacyclin synthase is one more enzyme that can convert our prostaglandin into prostacyclin, which is indicated as PGI2. This prostacyclin is converted into 6-keto PGF1 alpha in the endometrium. Whereas the coming to the HPETE, that is hydroperoxy ecostateroic acid, which is converted into leukotriene, that is A4, and the help of the water including it can be converted into LTB4. Also, the same leukotriene A4 can be converted into leukotriene C4, that is in the presence of glutathione. Hence, the enzyme is called as glutathione transferase. This leukotriene C4 is converted into leukotriene D4 with the help of the removal of glutamic acid. Hence, leukotriene E4 is formed. The thromboxane, which is formed from the prostacyclin, is converted into thromboxane TXA2. Different pathways of icosanoid synthesis are there. The first one is the cyclooxygenase pathway. The cyclooxygenase pathway forms all the icosanoid having ring structures such as prostaglandins and thromboxanes. The cyclooxygenase enzyme isoforms cyclooxygenase 1, which is abbreviated as COX1, and cyclooxygenase 2, that is COX2, are both implicated in this pathway. COX1 or COX2, no, COX1 or cyclooxygenase 1. Prostanoids are produced under physiological conditions by the COX1. The production of platelet aggregates, renal function, vascular homeostasis, and other typical operations are among the physiological cellular processes that COX1 regulates. COX2 or cyclooxygenase 2. In areas of chronic disease where there is inflammation, COX2 stimulates the formation of prostanoids. COX2, however, is constituently expressed in kidney, bone, and brain tissues. Lipoxygenase route. This pathway is responsible for the synthesis of lipoxygens and leukotrienes. The enzymes known as lipoxygenases reacts with arachidonic acid to produce unstable peroxide derivatives such as 5-HPETE, I2-HPETE, and I5-HPETE. These unstable chemicals can subsequently be transformed into leukotrienes, lipoxygens, or hydroxylated derivatives HETs. Asthma can be treated with anti-leukotriene medication. Coming to the summary, eicosanoids are lipid derivatives of unsaturated fatty acids with 20 carbons that have biological activity. Eicosanoids belong to the class of paracrine hormones and are bioactive hormones which local action that act close to the site of hormone synthesis illness. Arachidonic acid and related polyunsaturated fatty acids, including eicosapentaenoic acid, are the sources of eicosanoids. Four types of eicosanoids are present. Pathway cycle of COX1, COX2, lipoxygenase are also present. Thank you.